Hello, grade 10 learners. Today we will talk about activity 4 in chapter 1 that talks about photosynthesis gas exchange. As an introduction, photosynthesis is accompanied by gas exchange with the environment, the absorption of carbon dioxide and the release of oxygen. How can this gas exchange in green plants be proven? What is the source of carbon used for synthesis of organic materials? These are the posed problems that will be achieved by the experiments of the activity 4. According to the first paragraph, which deals with proving the release of oxygen by an aquatic green plant. It is on page 24 in your biology textbook. First, because it is easy to prove the gas production of a green plant in water, so we used an aquatic plant which is called Elodia. Document 1 shows the scientific or experimental setup that shows how Elodia proves the liberated gases as gas bubbles, shown in these figures. In the first setup, we introduced a large beaker in which we introduced a glass inverted funnel, and then a test tube is being inverted on this glass funnel. Okay, and we introduced the Elodia plant under these ones, and finally we immersed them all in water, so gas will be liberated and collected at the top of this test tube, which represents oxygen. According to what we studied in the previous lessons of the previous videos, we all know that photosynthesis takes place in green plants, but they have to use light with carbon dioxide for green plants in the presence of light to produce gas, which is oxygen gas, and starch inside its leaves. So by this experiment, the liberation of oxygen gas indicates that photosynthesis is taking place in these green plants. As a source of carbon dioxide, sodium bicarbonate produces carbon dioxide which is soluble in water, along with water which will be reacted with carbon dioxide in the green leaves, which will make photosynthesis to produce the oxygen gas in the presence of light. In parallel, we use the same setup but in an opaque box which provides darkness and prevents exposure to sunlight. What do you see in the inverted test tube? Is there any oxygen gas? No. There is no bubbles like the control one. So carbon dioxide is not being used, and photosynthesis consequently is not taking place in the second setup because of the absence of light, which is an important factor for photosynthesis. Okay, so the tested hypothesis of making this second setup is proving that light is an important factor for photosynthesis, because photosynthesis didn't take place in the absence of light. Then, in the third setup, light exists. However, carbon dioxide is not provided to the Elodia plants. Look, sodium bicarbonate is not introduced in the third tube. So gas bubbles are not being produced. So what do you conclude from the results of this setup? Exactly. Carbon dioxide is an important factor for photosynthesis. So what do we have to learn from this experiment? That oxygen also is gas liberated from photosynthesis, which constitutes the lungs of the whole environment of our Earth. That's why green plants constitute the lungs of our Earth, which produce carbon dioxide and consume which produce oxygen and consume carbon dioxide, the gas that we respire. Now let's move to document B in the same page 24. By this experiment, they were increasing or rising the intensity of light exposed to the green plants, and they recorded how this oxygen gas was liberated by these green plants. So as we see, as they increased the light intensity, and the rate of produced oxygen was increasing, indicating that the higher the light intensity exposed to green plants, the higher production of oxygen liberated by these green plants, and consequently more rate of photosynthesis takes place in higher intensity of light. Now, by taking an example on the effect of intensity of light on the rate of photosynthesis by a green plant, we considered exercise 6 on page 31 on your textbook of chapter 1. This study was showing that the graph below represents the variation in the volume of oxygen released by green algae exposed to light as a function of luminous intensity. The graph shows that as the intensity of luminosity or luminous intensity in the unit of lux was increasing from 0 up to 30 thousandths, the volume of oxygen released by the graph was increasing from 0 to reach its maximum of about 60 AU. However, as the intensity of luminosity or intensity of light increases higher than 30 thousandths to reach a value of about 33 thousandths lux, the volume of oxygen was decreasing to become 0. So what do you conclude? Exactly, we can conclude that luminous intensity is very effective and raises the intensity of photosynthesis in green plants to a certain extent. So the very high luminosity of light can destroy the ability of green plants to make photosynthesis. Okay? So مشان هيك نحن ما دايما بنقول انه كل ما بدنا نعلي الضوء لحتى تسرع اكتر البلانتس تعمل فوتوسينثيسيس، بيجي قوة معينة لما بتصير كتير قوية الضوء تاعو الشمس او اللمبة اللي نحن عم نحطها عليها للجرين بلانت، بتصير ما بتعمل oxygen و consequently يعني photosynthesis will not take place. مشان هيك قلنا انه الكلمة من قوة الضوء للبلانتس كرمال يعملوا مور photosynthesis هي مؤثرة وبتعليها لحد معين to a certain limit. مشان هيك قلنا بس to a certain extent. اوكي، مش انه يعني هيك كلمة بنعلي الضوء بتقوى الفوتوسينثيسيس. انتبهوا له، هذه ملاحظة كتير مهمة. On the other hand, exercise 9 in the same page 31 presents another idea concerning the color of the ray of light. يعني in this exercise they exposed to a radiation spectrum a spirogyra, which is a green algae filaments with a colony of aerobic bacteria. As observed under the microscope, the bacteria are found to be differently distributed around the spirogyra. Let's discuss the experiment before proceeding with the questions. In this experiment, they put a test tube in which a filament of spirogyra was introduced inside this tube. They put a red light on the left side, a green light on the middle side, and a violet light on the right side. Now, the upper band of the graph shows the absorption spectrum of chlorophyll, which indicates, so these bars with the highest frequency are observed with the red light rays, red light color يعني, whose wavelength ranges between 700, 600 and 700 nanometers, which is a property of light rays. However, very few number of absorption of light is observed in the green color light by the spirogyra. However, also moderate amount of absorption spectrum or absorption by the green plant occurs. يعني in other words، إذا بدنا نشرحهم أكتر، شوفوا كيف هون يعني السبايروجايرا هيدي البلانت عم تمتص أكتر شي ضوء لما بتكون عم يوصلها ضوء لونه أحمر، بتمتص أقل شي ضوء لما بيكون عم يجيلها الضوء الأخضر. والسبايروجايرا كمان هدول البلانت بتكون عم تمتص ضوء متوسط، مش كتير ولا قليل، بس يكون الضوء لونه فايولت اللي عم يجي لعندها. هلا in parallel إذا بتلاحظوا the bacterial colonies، يعني قديش عم يكبروا البكتيريا، قديش عم يتكاثروا، is very high near the spirogyral filaments when they are exposed to red light. يعني كانوا البكتيريا لأنهم هن aerobic بكتيريا، يعني بيتنفسوا، يعني بدهم أكسجين، كانوا هون أكتر شي عم يجيلهم أكسجين البكتيريا، فعم يتكاثروا أكتر شي. لاحظتوا كيف كانت very high. هون the bacterial colonies in the green light they were very few amount، كتير كانوا قليل البكتيريا، لايكو كيف نل تقريبا شحطة هيك مبين، لأنه الضوء خفيف بالنسبة للبلانتس، لأنه ما عم تقدر تمتص منيح ضوء، و consequently oxygen is not being produced in very high amount. Also light was in the violet light because لأنه السبايروجايرا كانت عم تمتص كتير لايت أكتر من الأخضر. The amount of bacterial colonies was high because the oxygen was being produced sufficiently or enough amount by the spirogyra, which is consequently making photosynthesis. So what do you conclude? We can conclude from this experiment that green plants can produce oxygen or the rate of photosynthesis by green plants is affected by the light color of the spectrum. يعني حسب الضوء اللي بيجيلها للجرين بلانت بتصير تعمل فوتوسينثيسيس. وكان هون الدليل عنا دليل انه كل ما عم يكتروا اكتر البكتيريا حدا يعني هي عم تنتج اكسجين اكتر. فليكوا كيف كانت سبايروجايرا بالاحمر عم تنتج كتير اكسجين فعم يتكتروا كتير حدا البكتيريا. كانت السبايروجايرا ما عم تنتج منيح اكسجين فقليل كتير حدا البكتيريا ما عم يقدروا يتكاثروا لانه قليل الاكسجين يعني consequently photosynthesis is not taking place in this region. However, in the region of violet ray light color, the amount of bacteria was high, indicating that oxygen is being produced and consequently carbon dioxide is absorbed and photosynthesis is taking place. Okay. يعني بهيدا الدراسة بتقولنا انه اللون الاخضر اللي هو مثل لون البكتيريا سوري اللون الاخضر مثل لون هذا الجرين بلانت ما بتستفيد منه كتير البلانت because it's being reflected. واحسن شيء لها هو الفايولت color light and red color light. اوكي تعال نجاوب هيك على الاسئلة لنشوف هن شو سائلين هون عن هذا الموضوع. Why do the bacteria approach the algae؟ انه ليه البكتيريا بيقربوا على الالجي؟ simply because algae are green plants which always produce oxygen and bacteria likes to survive in an environment that is rich in oxygen. طيب part 2. Would the bacteria behave in the same way in darkness؟ أكيد لا. Why? Because in darkness green plants will not produce any oxygen because photosynthesis will not take place. So bacteria will not approach the green plant. يعني لأنه بالعتم البكتيريا ما هي بدها تستفيد وبدها أكسجين. لما بتشوف إنه الجرين بلانت ما عم بتطلع أكسجين أكيد ما حتقرب لها. طيب البارت 3. What can be deduced from the different distribution of the bacteria؟ هذا اللي نحن شرحناه من شوي. بدنا نقول لهم إنه since the amount of bacteria was different in the different spectrum of light color, then green plants intensity of photosynthesis differs according to the light color it received. تعال نشوف الاكسرسايز 7 اللي هو page 31. بيقول لك هون the graph below represents the variation of volume of oxygen produced by an elodia twig placed in light as a function of amount يعني percentage of carbon dioxide present in water. شوف هذا الجراف. الجراف هاي عم تفرجينا لكن the variation of volume of oxygen released by the green plant which is elodia as a function of concentration of carbon dioxide. يعني نحن هون عم نشوف تأثير الكربون دايوكسيد كمية الكربون دايوكسيد الموجودة بالenvironment على rate of photosynthesis which is indicated by the volume of oxygen released by the green plant. According to this graph, we can see that the volume of oxygen released was strictly increasing as the concentration of carbon dioxide was increasing. So what do we conclude? We conclude that as we increase the amount of carbon dioxide, the rate of photosynthesis in green plants will rise. Okay? طيب هلا سألونا هون سؤال. Interpret the graph. The action verb which is interpret requires two elements: analysis and a conclusion. So first of all, let we analyze the given graph. Let's consider that the volume of released oxygen is being 30 AU. Okay, 30. No, it is 30. Okay, 10. So يلا هلا بدنا نبلش. We always begin with the results obtained or the dependent variable of the experiment. So هلا اللي بدنا نكتبه هو انه the volume of oxygen released by Elodia green plant increases from 0 to 30 AU as the concentration of carbon dioxide increases from 0 to reach 0.3%. هيك خلصت الاناليسيس. هلا significance لنجاوب على interpret بالكامل. This indicates that the higher the concentration of carbon dioxide introduced to a green plant, the higher the volume of oxygen released by this plant. So let's move to a part of critical thinking, which is part 2. They said that the value 0.03% of carbon dioxide represents its amount present in air. So how can the atmosphere of a greenhouse be enriched with carbon dioxide؟ يعني هون عم يقلنا إذا هيدي 0.03 هي كمية الكربون دايوكسيد that exists in the atmospheric air. So how can we enrich greenhouses with carbon dioxide? Simply we burn some dry leaves, and its burning will produce carbon dioxide inside the greenhouse. So the greenhouse will be enriched in carbon dioxide, and consequently the rate of photosynthesis will increase, and the productivity of these green plants will increase, and consequently oxygen being released of the green plants will rise again. Now moving to the third part of this exercise, they said, why do we say that green spaces are the lungs of the cities؟ ليه نحن بنعتبر the greenhouses are lungs of the cities? Simply because they produce oxygen. So as we enrich the environment of a city by carbon dioxide, green plants will consume them and produce oxygen gas in order to enrich the environment of a city with oxygen gas, and consequently a healthier environment will be for these cities. Now moving back to page 25 on your textbook, the second part of the activity, this activity 4, is dealing with proving the absorption of carbon dioxide by a green plant. So by this part of the activity, we will prove that green plants are producers of carbon dioxide, and they are respiring just like us. We all know by respiration we produce or liberate carbon dioxide and we consume oxygen by a process called respiration, which is the opposite process of photosynthesis that consumes carbon dioxide and release oxygen. To prove this idea, we used an indicator which is called cresol red. Cresol red is an indicator of the pH. It is pink in a natural medium, red in a basic medium, and yellow in an acidic medium. The carbon dioxide in the water makes the medium acidic. So the idea is, as green plants will produce carbon dioxide in a liquid, the carbon dioxide will react with H2O, which is the water, and will produce an acid. So this acid will turn the cresol red indicator into a yellow medium. Into the carbon dioxide and the water mix. Yes, into a yellow medium. اوكي، يعني الكربون دايوكسيد اللي بيطلع من البلانتس لما بيتحد مع المي هيصير اسيد، والاسيد هيحول لون الكريزول ريد ليصير لونه يلو. شوفوا كيف هون الكريزول ريد is a substance whose color is red. So in the first tube 1, they introduced a green living plant and enclosed it into a test tube with the space empty, and they introduced the cresol red at the bottom of this test tube. So a red color will be obtained by the green plant in test tube 1. However, in the second test tube containing the same setup, cresol red at the bottom of the tube with a green living plant and enclosed tightly, and this second tube was not exposed to light anymore. It was put in a dark opaque box, so light will not reach this tube. We can see that yellow color is obtained by the cresol red, indicating that an acid is being produced by the carbon dioxide released by the green leaf, and consequently it indicates that the green leaf is producing carbon dioxide, and proving that green plants also respire just like us. So green plants in darkness, they respire, and so the green plants in light will respire and undergo photosynthesis. Now the question is, why do green plants produce oxygen although they respire? Simply because the rate of photosynthesis by these green plants is higher than the rate of respiration. So the amount of oxygen consumed by these green plants is much less than that of oxygen released by photosynthesis by the same green plant. Finally, this experimental setup shows how the detailed structures are being introduced by this experimental setup. You should guess what this experiment means.